Today we will be discussing on hyperspectral remote sensing for agricultural applications. Yesterday we have been discussing on its potential on soil applications. So there is a big difference between the soil and vegetation with respect to hyperspectral remote sensing application is concerned, and this typical spectra is very much familiar with you all, as you have seen yesterday and also in the previous classes taken by other guest lecturers. So this shows the typical spectral pattern of a vegetation, how it looks like on different wavelength spectrum starting from 400 to 2500 nanometer. And here the purpose of showing this is how the unique interaction takes place at different spectral ranges, starting from visible to near infrared to sort of infrared range, and which is identified as a signature, and based on which you are able to identify the vegetation from soil and also from water, and again within the vegetation types how they are differing from each other. Then coming back to hyperspectral and multispectral, as you see over here you have 6 bands of Landsat TM showing the spectral pattern of a vegetation where which is indicated here as the blue spectra having 6 bands, 1, 2, 3, 4, 5, 7. Of course the 6th band is beyond the spectral range over here, so that's why there's a thermal band which is not shown over here. And compared to that, you could see the spectra given for in the green and yellow of a vegetation which is in different weathering conditions, and the red and green when you see and with the blue spectra you are able to see the difference. The difference is that when you move from 4th band to 5th band, there is a straight line coming in the blue. When you take in the multispectral and draw a trend line, or a straight line is in between, you don't have any information in between because you don't have the sensor. But when you have a hyperspectral data like a spectrometer data, Hyperion image, in that case a lot of up and down you are able to see. That's somewhere at 970, somewhere at 1280, somewhere around 1400. So that's little change in the composition of the vegetation, maybe the leaf water content or the cell structure. That kind of reflection you are able to see in those near infrared range, also in the visual range. So what I mean to say over here is, when you see the multispectral and hyperspectral, you are able to see it looks to be more informative compared to the multispectral, and little change in the spectral pattern, which is reflected because of unique interaction of the material, because of the change in the material conditions or the compositions, is reflected through hyperspectral reflectance. That's the reason hyperspectral reflectance can be potentially used to characterize a vegetation, to estimate the parameters, and also to know the process-based studies in case of vegetation. Now coming back to typical spectral reflectance of a curve, which is a very basic principle might have been taught to you through the basic principles of remote sensing courses, which is offered through EduShot again. And also maybe many people might have told you, you must be very familiar with this curve over here. It shows, you see, the visual range is more governed by pigments. The pigments are listed over here is chlorophyll A, B, beta carotene, xanthophyll. Many pigments are there which controls the visual range. That means little change in the chlorophyll content or beta carotene or any other pigment, that reflectance pattern is going to be changed over here in the visual range from 0.4 to 0.7. So that's what any pigment quantification you want to do, that can be done having hyperspectral in this range 0.4 to 0.7. When you come back from 0.7 to 1.4, that is in the near infrared range, that is again more because of the high scattering, because of spongy mesophyll cells, and also high internal reflection takes place. So you are able to see that kind of reflectance pattern, and which may be varying because of again the structural arrangement of cells, the cell wall compositions and all those things. So details we are going to discuss in the subsequent slides. So that's the reason you will be able to see some kind of you can characterize the cell structure, you can characterize the plant with respect to some of the water absorption band at 970 and all that, with respect to near infrared range. When you coming back to the SUI ranges, which starts from something around maybe 1.2 to up to 2.6, you see there are big dip points are there at 1.4 and 1.9, and that is because of water absorption bands, and which indirectly quantifies the leaf water content at equivalent water thickness in the plant. And of course, there are other parameters, other compositions of the vegetation or leaf which can be quantified, which you are not able to see that up and down in the reflectance curve, but you can quantify those if at all you analyze that using different approaches. Then coming back to the leaf biochemicals, how they affect the reflectance, you can look back to 1989, which is contributed by Dr. Kuran and Fortis et al. Later 1996, and also Lucas and Kuran 1999. And you can see the wavelength ranges, which is given here from 640 to 2350. And the biochemical parameter, one can quantify or go for studies on those, how they are changing with respect to leaf of a plant, or within different time of plant growth time or plant stages, how they are changing. One can estimate all these parameter using this wavelength ranges, which is very sensitive for that parameter, and the basic principles which governs the effect of biochemical parameter on this wavelength is given over here. That may be because of electron transition, maybe because of second overturn, maybe because of first overturn, maybe hydrogen stretches, NH stretches. So it all depends on what electromagnetic spectrum you are looking at and what kind of parameter you are looking at. So what I mean to say is, you are not only able to quantify these major governing parameters, that's a pigment or cell structure or water. Also you are able to quantify proteins, starch, cellulose, lignin, you name any pigment, any parameter which you are looking with respect to a physiologist or biochemistry, you can quantify using hyperspectral remote sensing. That's what it says, and we'll be subsequently discussing how this technique can be used to quantify those parameters. Now, looking at hyperspectral applications with respect to photosynthesis, which is a process which decides the life on the earth, you can see the basic equations of a photosynthesis, which is given by here, water with a carbon dioxide with a light, it synthesizes your carbohydrate and oxygen. And you can see this is the region which governs the chlorophyll content, or which is more governed by chlorophyll content. This is the region at which your energy get entrapped for the photosynthesis at 690 or 692, and based on which you are able to do the photosynthesis. So more the chlorophyll content, more potentially use your kitchen in the plant to assimilate water and carbon dioxide, which is again water absorption bands and carbon dioxide sensitive bands are here, which can be again quantified, and using them together you are able to get the carbohydrate and then oxygen measure. So what I mean to say is, whole hyperspectral remote sensing spectra or hyperspectral reflectance curve, if you can see from 400 to 2500, you are able to also see the process-based study like a photosynthesis. This is a very unique example which was sometimes back presented by Professor Robert Green from JPL lab, showing that how hyperspectral remote sensing can be used for process-based study like photosynthesis. Now coming back to the potential of hyperspectral remote sensing with the vegetation, these are the possible content which I am going to show over the time. What are the plant parameter estimation we can do by hyperspectral remote sensing? Mainly for monitoring, and that is maybe nutrient stress, water stress, that may be abiotic stress monitoring. Then coming back to the biotic stress monitoring, that is insect pest, disease or nematode. Then coming back to a concept called hyper BRDF that we are going to discuss in detail. Then how to discriminate different crops, also crop at different stages. If a crop is standing, then they're varietal, and how to characterize them in detail for different purpose. Then coming back to the approaches, what are the approaches can be used for estimation of all these parameters? Maybe based on simply spectral indices, maybe the spectral shift, that is Reddy's technique, maybe using those indices or sensitive bands through regression or empirical models by which you can predict those parameters, or through physical modeling like radiative transfer modeling, by which you can retrieve some of the parameters which is more of a physical based than the empirical based, both at the experimental level and also regional scale. Then of course for discrimination and characterization, or you have a spectra, how to know what kind of spectra, what is the target from which you got the spectra, That can be done by spectral unmixing or spectral matching. So all these techniques we are going to talk in detail in my subsequent deliberations. Now coming back to the abiotic stress, that's mostly nitrogen and nutrient and water. You can see here how there is a variation in spectral signature in wheat crop with varying nitrogen and irrigation in different phenostages. You see with the normal nitrogen and irrigation level, then this shows the different stage of a wheat crop standing. So you see wheat crop is standing on spectral reading when you take at different stages. You can see the spectral pattern how it happens to be. You have seen the typical spectra of vegetation, and this is what a typical spectra in the field having normal recommended doses of nitrogen and irrigation level, but at different stages of the crop. What I mean to say is here, having this spectra at different stages, you are able to discriminate, you are able to see that there is a change in the growth pattern of the crop at different phenostages, and subsequently you are able to get a result on the reflectance pattern, because of which there is also change in the spectral reflectance pattern at different stages. You see the CRA stage, tillering stage, booting stage, flowering stage and milking stage. So at CRA stage you will see kind of a crop growth is not that much as compared to your booting stage, which looks to be high reflection at the near infrared range and high absorption at the red band. And of course then next is the flowering stage, which comes down as the crop growth is the peak at booting stage, later it goes down. Then milking stage at which crop is going towards more of a senescence or maturity stage, so no more reflectance at that high at near infrared, or chlorophyll content won't be that high compared to your booting stage. So at different stages of the plant can be also captured by this reflectance pattern at different wave bands, and also you will be able to see using some of the sensitive wave bands, some of the sensitive spectral regions. You can also quantify at what stage is the crop. Then coming back to the optimum stage, that's what the booting stage. If you take a nitrogen treated plots having 0 kg, 30 kg, 60 kg, 120 kg hectare of nitrogen, then you can see the spectra, how they are behaving, how they are differing from each other. So a 0 kg nitrogen treated plot is almost a stress plant. It doesn't have a nitrogen content, as maybe the soil may have inherent nitrogen content, but before this wheat crop was taken up in the Kharif, the maize crop was taken, which was mainly nitrogen eater. It takes a lot of nitrogen from the field. So when you conduct a nitrogen related experiment, one has to take a Kharif crop as a maize, so that soil doesn't have much nitrogen now to feed to the second crop. So whatever the treatment you give with respect to nitrogen, the response is going to be good in that crop which you are looking at. So that's the reason now when you see 0, 30, 60, 120 kg nitrogen at the booting stage, you are able to see there is a differential spectral pattern with respect to different nitrogen treatment. Then coming back to different irrigation level, which was given based on the soil moisture deficit. One is the no irrigation is given into that plant, another is 30%, another is 60% of soil moisture deficit, another is 100% of soil moisture deficit. So based on this, you are able to see that there is also spectral pattern changes with respect to different kind of water stresses. So both the spectra shows there is an effect of nitrogen stress, there is a effect of water stress, and of course both are interrelated to each other, that we are going to discuss later in subsequent slides now. Now the first and foremost thing what we do is the correlation. Yesterday also I talked to you about organic carbon content estimation in the soil, having a spectral reflectance pattern, where the first thing what we did is correlation ranking, and also correlation plot we have drawn, and we could see what are the sensitive bands which is sensitive with respect to organic carbon. So same exercise can be done also in case of vegetation, where we could see the plant nitrogen accumulation, that is nothing but the amount of the nitrogen content in the plant in terms of concentration, plus how much is the biomass per unit area. So if you put them together, multiply them together, that is nothing but plant nitrogen accumulation. So you could see here the correlation, the relations between reflectance pattern of the plant nitrogen reflectance pattern in the all over the spectra starting from 350 to 2500, and the plant nitrogen accumulation. The spectral pattern in the tillering, booting and flowering stage you are able to see. So most sensitive stage, tillering stage, you are not able to see that proper pattern, but you see in the case of booting and flowering, you are able to see high correlation somewhere around nearby 700 ranges, sometimes around 1300 ranges, and of course you are getting a high correlation somewhere beyond 1800 up to 2500. So these are the spectrally sensitive bands which can be used for nitrogen stress monitoring. Then, as I told you, having those spectral sensitive bands are based on the basic principle on which the nitrogen content of the plant affects the chlorophyll content. Chlorophyll is the pigment which affects also your reflectance pattern in the vegetation condition, visible range. And so whatever reflectance you are getting in visible range is going to be affected by nitrogen content. That is one thing. Second, by the correlation ranking or correlation study, you are able to see also some of the sensitive bands which is beyond also visible range. So there are many authors who have published so many work on using these sensitive bands, different indices they have developed, on which not only they could see the nitrogen monitoring, also crop growth parameter, for example, leaf area index, above biomass, and also chlorophyll content. So these are the indices. The simple ratio, which is hyperspectral indices, simple ratio has taken as reflectance at 900 and reflectance 680, the ratio of that. There is another index called photochemical reflectance index, that is PRI, which is taken at reflectance 550 and 530, the normalized index again. Then hyperspectral normalized difference vegetation index. This is the simply NDV index. The broadband has been taken here at particular waveband, that is taken here, which is developed by Penulas, then later by Sims and Gammons. Then red edge value, red edge technique, that we are going to discuss much in detail, later we will be discussing. Then modified chlorophyll absorption ratio index, again at specific bands it has been taken, the expression is given over here, which is very sensitive with respect to chlorophyll content, given by Daughtry et al. Like this, those indices are there which are very specific with respect to the parameter you are looking at and could give very good result with respect to the leaf area index or chlorophyll content. You can see here, like this we have evaluated all those indices. For example, one index is given over here, is how this seasonal pattern of index is affected by nitrogen fertilizer treatment. For example, physiological ratio index, which I have shown you over here, is 550 and 530. This is the normalized one, and you can see here, down here the date of sowing is given at different phenostages. We have taken the spectral data. The blue one shows the plot which was having zero nitrogen applications, and N1 is the 30 kg, N2 is 60 kg, N3 is 120 kg. So you can see, using this index, which looks to be very sensitive with respect to chlorophyll content, you are able to see how the temporal pattern of the PRA, from which you can know which plant is behaving behaving good with respect to growth, which is bad. That means nitrogen monitoring can be done also using these indices as a quick monitoring system. So what you can see over here is, as you see the temporal pattern, the booting stage, that is sometimes around 70 degrees after sowing, 70 days after sowing, looks to be optimum, at which the PRA attains also optimum. That's the best stage of the crop, at which all the biomass attains the peak, Li attains the peak, and also same also with respect to the reflectance which you get or indices which you get, and that's what you are able to see very clearly over here. And initial stages, you are not able to, since the crop growth was not much and nitrogen treated, of course all the stages you are able to see differential behavior of this PRA pattern, but you are able to see that at different stages the nitrogen stress monitoring can be done using indices, something like one example is PRI. Using these indices, we could develop some of the models, like here you have given those models like simple ratio, HNDVI, PRI, soil adjusted vegetation index. All those indices are given here, and different prediction models, empirical approach was used to develop these models, and those are evaluated with respect to LI, leaf area index. And you could see here what is the R-square value. Almost the R-square is very significant and having 0.83, 0.79, 0.69 and 0.86 using different indices. And you can see also error term or RMAC, which also tells you how good is your prediction model is. So what I mean to say is, you can use these indices for biophysical parameter estimation through simply empirical approach also. So the biophysical parameter here is Li, and the indices which you have used is simple ratio index, HNDVI, PRA, and all. So from this one can choose which index should be taken for the parameter. Any of these you can use for estimation of Li. Then for the chlorophyll, again some of those indices have been used, and the prediction model was developed and they are evaluated with respect to the measured and predicted. You can see 1:1 line how these indexes were predicting agreeable results with respect to ASCA value 0.9, 0.91, and 0.82. So these indices also can be used for estimating chlorophyll content in the plant. So before going further for the nitrogen stress detection using hyperspectral remote sensing, we have been till now what I have discussed is the sensitive bands, then their derived indices. Now there is another technique called red edge technique. What is that is, when you go from red band down here, when you see the left hand side, the curve given over there, when you go from the red wave band to the near infrared, all of a sudden the reflectance shoots up. You can see the 0 kg, 30 kg, 60 kg, 120 kg treated plants, hardly there is any change with respect to that slope curve which you see over here. Hardly there is any change. You can't see here anything visibly difference with respect to these treatments, but you are able to see all these cases. But when you take the first derivative of them, as yesterday I discussed, if you take the mathematical transformation with respect to how much is that slope change of the reflectance, or how much is the change in the reflectance with respect to little change in the wavelength, if you take that, that's what is given over here. There is the first derivative of reflectance. If I take that, that is nothing but del R divided by del lambda. If I take that, you are able to see here, it is almost a straight line, and there is a peak over here which is because of this change in the slope. Again, from 680 to around 750, which is the range from here to here, 680 to 750, from here 680 and 780, you are able to see there is a difference which is very much visible with respect to these treatments. So same thing is magnified over here. So if you take a first derivative of this reflectance, you are able to see some information which was not able to before, which you are not able to retrieve or interpret any information out of that. But now when you see the first derivative result, what you can see is there are two things. One is with respect to nitrogen treated plants like 0 kg, 30 kg, 60 kg and 120 kg, the value, the inflection point, or the red edge value, we call it the red edge value, that is the perpendicular height from that wavelength, from the baseline. What is that height? This height is called the red edge value. So that red edge value is changing significantly if you go from one treated plant to another treated plant, from 0 to 120 kg, and they are directly proportional to each other. That means more the plant is, if plant is healthy with respect to nitrogen, higher the red edge value will be. That means the slip, the gradient will be high. The change in reflectance with respect to wavelength will be very high. That's what it shows. That's the first thing. Second is, if you look at a healthy plant and a stress plant, healthy plant is your 120 kg nitrogen treated plant, and a stress plant is 0 kg one, which is nitrogen stress plant. You see in both the cases what happens. One thing is your red edge value is changing. Another is that peak position is shifting towards the lower wavelength. So it is moving like this. So when you go from a healthy plant slowly to the stress plant, there is a change in nitrogen stress, a change in the peak value. So that tells you how the plant is behaving and how the stress behavior is. So if there is a shifting towards the lower wavelength range, that's called blue shift, plant is under stress. If plant is under stress and you treated that with the fertilizer applications, if it goes for a healthy one, it goes the red edge position, the red edge position goes towards a higher value, that is called red shift, plant is in healthy condition. That means if you take this red edge position and red edge value, one can quickly monitor the nitrogen stress in case of plant. So that's what the message goes from here. Not only based on the indices or sensitive bands, also one can look at this technique called radius technique for nitrogen stress monitoring. Same thing also can be used for disease and pest monitoring. That's what we are going to show you in some of the slides. So not only you can monitor this radius, the nitrogen stress by radius technique, also you can monitor the stages at which the plant is. For example, you see over here, the CRA stage, you see the radius value is very less. Then in tillering, the radius value is increasing. Then in booting, almost it attains the highest value. Booting is the best stage at which it attains the highest value. Then after booting, in the flowering it comes down, then milking it comes down. As I told you, booting is the highest, the peak growth stage. After that the senescence starts. That's why the radius value goes down. So that means you can know the crop growth stage if at all you have a radius value or radius position. Radius value is that perpendicular height. Radius position is where is that peak lies with respect to the wavelength. So having these two parameters, not only you are able to monitor the nitrogen stress, also you are able to know what stage is plant. And this technique is mostly used in case of forestry, in case of forest plants, trees. What is the stage of the plant, or how mature is the plant, what age is the plant. The aging of the plant, the tree, is done using this technique. Then different indices with respect to radius value is compared. You can see here with respect to the leaf nitrogen content, there are good prediction equations were developed having R-square value something above 0.8 and different indices, which looks to be good with respect to nitrogen estimations, and I have shown you already the validated result of those. And so taking DRI is nothing but the radius value, that is the derivative with respect to the change in the reflectance with respect to the wavelength. So if you have a radius value on the y-axis, which is a function of nitrogen, one can use this radius relationship with respect to nitrogen level, and also can go for fertilizer applications using these prediction equations. So if at all you know the stage of the crop, you know how much stress is having this radius value, from there one can look for how much nitrogen can be applied. Then coming back to plant nitrogen accumulation with respect to different indices which are very sensitive with respect to nitrogen content, like there are some of the indices given over here. You can see the prediction model with respect to observed one, how good they were. So nitrogen stress can be very well monitored with respect to using this hyperspectral remote sense technique. Not only nitrogen, phosphorus, sulfur and potassium can be done very well also. Now coming back to water, similarly, like in case of nitrogen, you could see some of the sensitive bands which are used for nitrogen stress monitoring. Similarly, having the correlation ranking studies, one can look for the indices, for example moisture stress index, where it is nothing but the ratio of reflectance at 1600 by reflectance at 820. 820, there is hardly any change with respect to the reflectance with respect to different water content, but at 1600 there will be significant change with respect to moisture stress. Same also with respect to water band index, where 900 by 970 is the water absorption bands. So using those sensitive water absorption bands, these indices have been generated and can be related to the leaf water content to know how much is the water stress, or to know the leaf water content. For example, you can see over here, you have a saturated plant and you have a highly stressed plant. This is what the saturated plant, T0, and T4, the upper one, is a highly stressed plant. As I have told you before, showing you typical spectra of vegetation, starting from 1000 up to 2500, the spectral pattern is morely governed by water stress or leaf water content. So that's why you see there are drop downs at here, here, here and here at these points at 970, 1280, 1400 and also 1900. These are very unique water absorption bands at which one can look for how much is the water content in the leaf, or leaf water content can be measured. And that's the basis on which all these indices has been generated and related to leaf water content. So you can see here different sensitive bands taken 950 to 970, 1150 to 1260, 1450, 1950 and 2250, and at different stages the relative water content prediction equation was developed and R square value was measured, and you can see in the British stage the relatively prediction equation was found to be very good, and you can see here the relationship how they are with respect to different wavelength ranges and relative water content. Then coming back to biotic stress, which is mainly because of the insect, pest or diseases, you can look at here the spectral pattern of a rice crop heavily infested by rice leaf folder during Kharif 2007. So this is the field at which there is a different level of infestation by the leaf folder, which was done by the scientists from the Department of Division of Entomology of the institute. Then here we are taking the spectral observations having the severity of the plant and different treatments, and this is the leaf folder you are able to see there, which damages the leaf and folds the leaf, and you are able to see the damage percentage given over here in different color in the lesions and the spectral pattern. So this could be also using different indices, one can quantify, differentiate how much is the damage is done and how much is the severity. But here the analysis is done only after it affected, not before that. Means after it's only post-mortem analysis. Once the insect is there, it got damaged. Once the plant is damaged, that kind of reflection, whatever you are getting through the instrument or the sensor, you are able to see that how much severe is the damage. But you can't predict the damage. That's another story which one has to look for. Then also another study was done for the brown plant hopper, which is also another important disease in case of rice. You can see the severity level which is scaled from 0 to 9, and you see the spectral pattern how they're really varying, having the plant at different severity level, having the nitrogen, water stress, everything, water content, everything controlled. Only this brown plant hopper damage level is varying 0 to 9, and you can see how they are varying. So the basic principles or etiology of the insect pest disease, one can understand what that insect does. It may damage the pigment content of the leaf. It may suck the cell shaft. The cell structure may get damaged. So which is reflected over here at the red band itself. Maybe the chlorophyll content may goes down, so the reflection will be high in case of red band. When you come back to the near infrared band, what happens is, since the cell structure is damaged, cell shaft is gone, so the cell may shrink, so it may affect to great extent the reflectance at the near infrared range and also at other ranges like maybe in the SUIR 1 and SUIR 2. And you could see here at somewhere around 1400, 1900 there is a gap given, as you might be knowing, using the field spectrometer. When you go to the field at 1400, 1900 you have the noise bands, so you get the spikes. When you see previous slides, these are the noise bands at 1400, 1900 which can't be used. These are the water vapor bands, and here also. So this has to be removed, and rest the spectral pattern has to be analyzed with respect to the parameter which you are looking at. That's the region which has been removed over here. And when you integrate those bands with respect to different ranges of electromagnetic spectrum, maybe UV, visible, blue, green, ray, violet, blue, green, yellow, orange, red, near infrared and short wave infrared. Those ranges are given over here. You are able to see how sensitive is the severity level with respect to reflectance pattern. And mostly you can see here the sensitivity is more in the near infrared range than any other bands. That's what you could see over here also. Same thing is seen over here. That means one can integrate those bands to the satellite level, maybe the MODI satellite or LIS 3 or LIS 4 or any satellite you have, multispectral satellite. One can look for whether this damage assessment can be done in the remote sensing satellite level in a regional scale. This is a field experiment which can be upscaled to the satellite level using these basic studies through hyperspectral remote sensing studies. Then using those as I told you, we have done some kind of a collision analysis also in this case, sensitivity of the different spectral regions with respect to the disease severity. Then we could identify some of the spectral bands which is very sensitive. Then prediction model was developed using those sensitive bands. This is the prediction model, and the right hand side is given using multiple regression models, and you can see 1:1 fit line how it is, and left hand side those three these three graphs shows how is the prediction model how is that prediction model is working with respect to BPS severity 1:1 line. Then coming back to the disease damage with respect to hyperspectral remote sensing, here again the yellow mosaic virus disease in case of soybean crop, which is shown here. This is a plant which is very healthy. This is a plant which is severely affected by the disease of the yellow mosaic virus. So the severity level is classified or labeled them from 1, 2, 3, 4, 5, and you can similarly you will get the different spectral pattern. And this is again, you can see here how severe chlorosis takes place. From green to yellow it has happened. So total chlorophyll content goes down, and carotenoid also get affected. So which is reflected in case of reflectance pattern, which is taken after 20 days after inoculation and 60 days after inoculation. So you have a disease level and a healthy plant, and if you take the difference of that, you are able to see that difference curve is given over here. You are able to see the difference is more here because of chlorosis, and chlorosis is nothing but the change in the pigment content, chlorophyll content, which is more governed at the visual range, and that's the reason you are able to capture this reflectance, the change in the reflectance at this visual range, which is shown over here. And using same radius technique, one can look for how severity the disease or whether the plant is under stress or not, and one can, using a ratio index simply at 750 and 445, you can develop a prediction model how disease severity is. What I mean to say is, also using hyperspectral remote sensing, one can look for disease severity. But here another challenge is, if there is a nitrogen stress, then plant also looks yellow. If there is a waterlogging, also soybean crops looks to be yellow. When you look at the major growing regions of soybean in the somewhere in the MP, in those regions, so you can see at a stress these yellow patches are there, and that may be because of waterlogging for a long time, or maybe because of nitrogen stress also. So that's the again another point, a researchable issue, which one has to see how using hyperspectral remote sensing one can differentiate whether it's because of the disease, that is yellow mosaic virus, or whether because of nitrogen stress or because of waterlogging conditions in the field condition. So one has to look for that. Then again there is a mango malformation which is done by there is one mango, what do you call that? There is a wilt disease, which is fungal disease, which causes the mango malformation, and which greatly affects the mango production in case of largely in India, which is mainly affected by relative humidity and also temperature. So one study was conducted in the field itself in the mango orchards, taking again the spectra to the height of the canopy and taking the spectra of malformed flower and healthy flower. And you can see here what happens in the case of malformed flower is it remains more of a greeny and biomass is heavy, and where in case of healthy flower, the healthy flower turns to red or pinkish. So that's the basic difference between them, and that's what the spectral pattern you are able to see here. But when you see the radius technique, if you take the taking from 680 to 720, you see the radius technique. What happens is, in case of healthy plant, there is a blue shifting, and as I told you, in case of blue shift, there is a stress in the plant. But in this case, blue shift is taking place in case of healthy flower, because healthy flower in the sensor looks to be stressed because it doesn't look green more. It doesn't have more chlorophyll content. It's more into pinkish color, which is mainly which mainly happens in case of a crop, a leaf, because of the chlorosis, the leaf, the green color changes to yellow or some other color. So here the case is opposite. So that's why what happens is, if there is a blue shift, that means plant is under healthy condition. If there is a red shift, plant is under malformed condition, so plant may not yield good mango. So that's what the interpretation in this case. There has also a study has been done by Bhattacharya et al. From Space Application Center using hyperspectral remote sensing data, that is Hyperion sensor, which detail yesterday I discussed with you. Two diseases they are able to discriminate using. One is sclerotonia rot, another is alternative blight. You are able to see the spectral pattern of different severity of alternative blight and also different severity of sclerotonia rot, and the indices were generated using sensitive bands for both the disease, and the mapping was done for where are the areas where disease is affected, where are the healthy in case of mustard plant. Then coming back to spectral discrimination or the estimation of some of the pigments like cellulose or lignin, this is the spectral pattern of residues of pigeon pea, groundnut, soybean, cotton, maize, and this is a pure soil. You can see the spectral pattern how what happens after the crop is harvested. The residue which is lying over the land there, that spectra was taken. It's more of a non-pigmented vegetation spectra, which doesn't look like a vegetation spectra as you see here, because there is no more chlorophyll content. It's almost a dried one, so you see the spectral pattern. But the difference, the message is here there is a differential spectral pattern with respect to different residues. So having this spectra, one can quantify also what is the lignin cellulose content at in all these residues. Looking at these wave bands, one can quantify cellulose absorption index, also lignin cellulose absorption index, which can be used in a spatial scale carbon sequestration. Like if when the plant is harvested, a residue is there, how much that residue is sequestered to in terms of carbon. So that can be done, whatever the sequestration model you use. In that case, one can enforce these indices CAI, LCA, which is derived again from hyperspectral remote sensing data or the reflectance, the bands, which can be enforced to have that model to know how much is carbon sequestered in a regional scale. Then looking at the tomato, how good is the tomato fruit, and or what stage is the tomato plant. If you have a hyperspectral reflectance curve, from there you can guess what stage is the plant. And the purpose here is, you can see here there are different stages of the tomato fruit: immature, green mature, breaker, timing, light red, red, and red ripe, which is the different stage of the tomato. And you can see the spectral pattern of the tomato fruit. What it indicates is, if you divide them into three regions like visible, near infrared, and SUI ranges, and you look at the visible ranges, what's happening is all this pattern of the tomato in different stages is governed by all these pigments, all these biochemical parameters. For example, phenols, lycopene, chlorophyll, carotenoids. So these are some of the sensitive bands which is governed by all these pigments. So if at all you have the spectra, from there you can know these pigments, and from there you can know what stage is the plant, the fruit is, whether fruit is, how the pigment transformation takes place from one stage to another stage, from immature to green mature to breaker to tawning, or light red to red. Or else having that sensor also in the storage, when the tomato is stored, whether the tomato is in a good stage to go to the market to fetch a good price. So all those studies can be, all those applications can be done using this hyperspectral remote sensing technique. Then weed discrimination. In case of precision farming, there are some sensors people have developed using this hyperspectral basic studies, sensors for weed discrimination. You can see here, these are some of the sensors. Yes, these are some of the weeds which is mainly dominate over wheat crop. This is a wheat crop with a nitrogen treatment, and these are the weeds. So you can see the spectral pattern of weeds, how they are differing. And here what it tells you is, this is the weed called chenopodium, which is with a different density in a wheat crop. Wheat crop is there with that chenopodium density. Density means that 8 plants per meter, 16 plants per meter, 32 plants per meter, 64, 128 plants per meter square. Means number of plants has gone up, the wheat plants has gone up, having the same wheat plants. So you are able to see the spectral behavior is changing. So if you have a spectra like this, later I am going to discuss, from there one can know how much percentage is the wheat crop, how much percentage is the wheat crop. From there one can threshold how much percentage wheat can be allowed in a crop standing so that my economic yield is not affected. Then also you can look for, within a wheat crop you have varieties. We have done some studies having 80 genotypes grown in the field, and these are the spectra of those different varieties. You can see the spectral pattern, how they are changing. Now we are in the process of discriminating how they can be discriminated so that we can know how genotypically these varieties are different, or how spectrally, if at all you have a plant standing, you can within no time you should be able to differentiate them, what kind of variety it is, or whether they are of same variety or different variety. Then looking at different rice genotypes of different stress level, again you can see here these are 18 rice genotypes. Of course it's not maybe may not be visual to you, maybe in the later slide I will be able to tell you. These are 18 rice genotypes which has been chosen to understand which is tolerant with respect to water stress. So this is a saturated condition, there is no water stress, and this is after maybe 1 hour, after 2 hours, after 3 hours, after 4 hours of the saturations. So over the time the water stress level has gone up, and that's the reason you are able to see there is a spectral change from near infrared down to 2500 nanometer. You are able to see here the difference, and that is because of change in the water level. Same thing shown in much in clear way over here is 6%, 16%, 26%, 48%, 90%. So you see here, this is the 90% is the relative water content. Here is the 6% is the relative water content. So the specific water bands, you are able to see the significant change in the water content. So having these bands, one can derive some indices. One can look at this depth. What depth is the water absorption? Means more the depth, more will be the water absorption. More is the healthy is the plant, or more is the relative water content. One can also look at the symmetry of the area of this this area, how much is the area which is got affected because of high water content, these water absorption bands. So having all these things, one can relate with the relative water content and develop some of the prediction equations. For example, you can see how different rice genotypes they behave with respect to water stress. So this is only conventional approach, 1 hour, 2 hour, 3 hour, 4 hour after the full turgid conditions. You can see relative water content with respect to different genotypes. So now look at the spectra using the indices, moisture stress index. Almost the same pattern you are able to see over here, but one thing you can see is how the sensitive is that genotype with respect to water stress. For example, you see here this and this. So this is at the saturated condition, this is at the stress condition. T4 is the stress condition. That difference tells you how sensitive is that genotype is. But for example, you see here, this is at the different stage how the genotype is behaving to the stress or to the water stress. Change in moisture stress index with respect to time, how they are changing, you are able to see over here. So what you can within no time you can see is that this is the genotype which is very resistant with respect to the stress. This is the genotype which is very sensitive with respect to water stress. Same thing is seen over here. If you look the sensitivity with respect to moisture stress index, we can choose the genotype which is highly resistant with respect to drought or water stress. So out of these 18 genotypes, one can choose having the low value of sensitive index, which is good for drought prevalent regions to grow that variety. So having those sensitive bands, you can develop the prediction models. This is what the prediction models were used: water band index, moisture stress index, NDII, NDWI, and these are the R-square value, and this is the what you could see. What happens is after 60% of relative water content, hardly there is any change with respect to the, there is no sensitivity with respect to these indices and relative water content. Of course, after 60% of relative water content, plant is almost, the 50% yield gets affected after 60% of relative water content. So nobody will wait till the plant attains to 60% relative water content. So what I mean to say is, So beyond this, it's not sensitive. So having a correlation analysis also, one can look at these sensitive bands. As I told you, 1450, 1780, 1930, those are very sensitive with respect to water absorption bands. You can develop the prediction equations quickly. And so in situ, relative water content measurement can be done having these sensitive bands only, not the whole spectrum. That's where the masses goes. As I told you, if you can go for continuum analysis, like how is that the pattern of the, how is that, what is the depth of the absorption water absorption band at particular wavelength range, one can look for continuous removal analysis or continuous removal factor. For example, you can see over here, this is what the continuous removal spectra is done. That means if this spectra is converted, this solder you join them and make it as 1, then this is something less than 1, this depth is taken, and based on that one can, based on the depth one can relate to relative water content, can quantify how much is the relative water content. And having this depth, one not only calculate the depth which is given over here at a particular wavelength, also you can know how much is the, what is the width and how much is the area and how asymmetry is this curve at that absorption band, unique absorption band. So based on this one can estimate, for example, water content, also chlorophyll content, or anything you are looking at particular at that absorption band. So these are some of the techniques which can be used using those water absorption or the specific absorption bands for estimating different parameters. So yesterday I discussed about continuous removal factor. Using this continuous removal factor, one can also estimate the relative water content. This is what the continuous removal factor which has been used and having a good R-square value for the prediction models. Then at the last, let's look for hyper BIDF, that is nothing but hyperspectral bidirectional reflectance fraction study, which looks to be very important with respect to vegetation. The basic principles over here is that nothing in this earth is isotropic in nature with respect to radiation as far as the radiation is concerned. For example, you see down here the lawn where in different directions you have the camera. This camera is nothing but the blue arrow, and source of light is given here as the sun. So you take in different directions the photograph, the lawn picture is going to be different. Why? The source is same, but the sensor in which the radiation is received is going to be different. So that reveals how anisotropy is any earthly feature is. So the relative position of sun and the sun or observer or sensor matters a lot with respect to the what parameter you are studying or what kind of radiations you are going to, how the radiation is getting affected with respect to the plant parameters. So that's the reason why one can look at a particular directions with respect to the source. For example, source is here, the light is falling down on the target down here, and the sensor is here. So in a particular directions, from this direction the light is falling down to the target and the other directions is going back. So that's why we call it bidirectional reflectance fraction, and this is quantified in terms of zenith and azimuth angle. The sensor position or source position is given by zenith and azimuth. Zenith is how much it is deviating from the normal, like from here how much is deviating, that is nothing but the zenith. Like if the sensor is just on the top of you, that is 0 degree, just above Nadi, that is 0 degree. Or if it is deviating like this, slowly from 12 noon till 4 o'clock you come down, the sensor, the sun goes down, so zenith is going to change. So that's what the zenith stands for. How much is deviating from the perpendicular line over here. And azimuth is, if you draw a line from up to this plane, then the angle between the north and this line, whatever the angle it makes, that is your azimuth angle. Means on which direction, whatever the source or sensor, whatever you are looking at, its position with respect to north is called the azimuth angle. So BIDF can be used to know the anisotropic nature of the feature. From there one can understand at what directions my sensor should be there, so that I can better correlate with the biophysical parameter I am looking at, or I can better understand the plant or target I am looking at. That's the purpose why BIDF is there. Then there are many ready to transfer models. Till now, what I have told you is empirical models, that's simply regression based approach on which you are able to get some parameters estimated. But regression approach, as you know, those are very subjective. In the equation which I have shown you, that may be working good for this year, may not be working good the next year. That equation may vary from again the experiment to experiment and doesn't explain scientifically any logic behind why those bands has to be taken. Only simply statistical model which was used to have those prediction equations. But in case of ready to transform model, what is used is all the physical understanding is done, how radiation is coming from source to the target, how it interacts with the target means the plant, interacts and goes down to the soil, again reflecting back to the canopy again and goes to the sensor. All those basic principles are understood, and based on that one can look for parameter estimation. For example, you see here, for example, canopy parameter you have, maybe so many parameters, chlorophyll or water content or leaf area index. Then you have a ready to transfer model. As I told you, having those parameters, I can model how much radiation I am going to receive. So if at all I can use those model, reflectance model, I am able to simulate my reflectance. That is nothing but the calculated reflectance R. So having this ready to transfer model, I am able to simulate. Same time I have a instrument, that instrument may be spectrometer, or I have an image from which I have a measured reflectance. So this measured reflectance and simulated reflectance, if you take the difference, you will get a del R. So if you try to, always you will try to make the del R to be 0, that means the simulated spectra should be exactly equal to measured one, provided your model is good. So that sensitive analysis mostly we do to make this del R 0, so that the model is now perfect to simulate exactly what I am measuring. So this is what you call is forward modeling or direct modeling, direct method. Now, once I have developed this model, once I have optimized the model, once sensitive analysis is done using making this del R equal to 0, now I can invert this model. What I can do is, now if at all I have the measured spectra, then I can invert this model to know what is my parameter which I am looking for. Maybe leaf area index, maybe water content, maybe chlorophyll. I can estimate that. So that's called inversion of the model. So you have a kinetic parameter measured C, and you have predicted now, that is C prime. You make the difference, that will be del C. So then del C you can make it to 0. If at all you can optimize your inversion technique. There are different inversion techniques which can be used to invert this ready to transfer model to retrieve the parameter. This is what whole concept is. And to understand the BIDF, so you have to put the sensor in different directions, for which you need a facility called goniometer. So this is what the goniometer is. In different directions, this is what the zenith position can be changed from 0 to 90 degree down here, and this is the azimuth position which can be changed. So this is what the goniometer is, on which this is a platform on which the sensor can be put in, and here the sensor can be put here. This is what the spectrometer, and the sensor can be put from 0 to 1 to like that. You can keep on changing at every 1 degree up to 90 degree or maximum up to 60 degree. 90 degree it look never to the target, it will look like this. Then you can rotate this arc over this with respect to sun or with respect to north directions, whatever you like to. If at all you look at a relative azimuth, then you can, relative azimuth means with respect to the source, sun, you can put the sensor making it a 0, the azimuth of the sun and source will be 0. Then you can keep on rotating so that at different azimuth your spectra you can record. So this is called goniometer, which is given again here. So this is what I was telling to you. So you have 0 degree, 20 degree, 30 degree, 40 degree, 50 degree, 60 degree zenith, and this is your azimuth, and this is what the sun position. So having these all possible combinations, one can take the data and look at what particular directions my radiation is going to be better. For example, you can see here a particular band. This study is the result is given over here. You see the reflectance from 0.34 to 0.48. So the red is high value, and you can see here this is point at which you get the high value. So this is called polar plot. So
you can look at which point, at what position of the sensor you are getting the highest value. That is somewhere around 40 degree you are getting. Then somewhere around relative azimuth 0. That means if the source and sensor, they are in the same directions, relative azimuth 0 means relative azimuth of the source and relative azimuth of sensor, they are going to be, the difference between them is going to be 0. That means they are almost maintaining the parallax. So that position, again, zenith, if you look at the, when the data was observed, if you look for the solar geometry, then zenith of the sun at that time was almost 40 or 45 degree. So that's the reason why you are getting the high value over here. So this tells you how anisotropy is a plant. That plant may be wheat or maize or anything. So that difference you are able to see over here. So this position at which you are getting highest value is called the Hotspot position. So one can advise this is the position at which you should put a sensor or source with relative to the source you should put a sensor to retrieve the parameter best. So same study was also
when you is that is in the field itself. Again, same was done in a regional scale, having the spectrometer down there and also measuring your different plant parameters. For example, chlorophyll content, canopy using canopy analyzer LAI. So different parameters were taken and 190 locations were visited, and the prediction ready to transfer model was used to retrieve some of these parameters in a regional scale. And the ready to transfer model which was used here was the ProCell model, which tells you, for example, these are the parameters which can be given into the model, that's the prospect model, and these are the parameters which can be given into the cell model. Putting them together, you can simulate a spectra, reflectance spectra. So this is all ready to transfer model. So you have N is the leaf structure parameter, this is the chlorophyll content, this is the water content, equivalent water thicknesses, and this is the dry matter content. This is the source and sensor geometry parameter and leaf area index and soil background, which can be taken in the cell. So putting all them together, you can simulate the spectra, what you can get in the spectrometer. So the optimization you can do so that the measured spectra and simulated spectra looks to be same. And the sensitivity analysis, you can see here how different parameters they are behaving different. As you see the chlorophyll content, the most variation, mostly the reflectance pattern is governed in visual range is mostly by chlorophyll, and this region is mostly by water. So all those parameters, the sensitive analysis was done, and then model was run to simulate the spectra, and observed spectra was evaluated, and you can see the pink and blue, the observed and simulated spectra, how they are matching to each other, and RMSE is given up for different bands. Once you could optimize the model, then one can invert that model to retrieve the parameter. Before that, as I told you, the sensitive analysis can be done. For example, the chlorophyll content is given in figure A, means as you change and keep on changing the chlorophyll content in the model, you see the spectra how they are changing. Only visible range the spectra is changing. Same also in case of water. If you keep on changing the leaf water content, then the spectra will be changing is starting from 1000 to 2500, not in visible range. So the sensitive analysis is done, then model inversion is done as like this. So these are the parameters which can be given into the model. So this is what the ProCell reflectance model. Then one can invert, one can get the spectra. You can simulate the spectra. So this is what the ready to transfer model is. But I want the parameter to be estimated. So what I need to do is, once my model is optimized, I can invert the model. Then I have the spectra observed, spectra by the instrument, or I have a image. Then I can invert the model, reflectance model, and can retrieve this parameter. So these are the parameters. Then same parameter you can evaluate how my inversion model is, as you measure in the field chlorophyll content, the leaf water content, LI by using some of the conventional techniques. You can evaluate how good is my inversion model is. Then having that, so this is what the model could be used to have leaf area index generated for a regional scale. That is a trans-Gangetic plain regions having Punjab, Haryana, and 2 districts of Rajasthan together for wheat crop. This is the wheat mask, and this is equivalent water thickness, and this is the chlorophyll content. So spatially you are able to see the Li product, you are able to equivalent water thickness product, you are having chlorophyll product. So which the NASA is doing, in NASA MODIS data they use, having they put in the public domain LI product so that you can use directly the LI product for any process study, either yield predictions or any study you want to do. So same also, for example, this map can be used for water stress monitoring. This can be used for biomass monitoring or yield monitoring. This can be used for chlorophyll, can be used for nitrogen monitoring. So the special variable maps, they can be used directly for site-specific nutrient management, water management, or yield predictions. And how good is that reduced model is given over here. Estimated LI and measured LI. Almost 90 points are taken here to evaluate that. And you can see here, over here. So in one advantage is here is that you don't need to have a raw satellite data to come out with this product. That is one. Second is the model which you have used is a very generic model. So once you have a satellite data, you can develop this product and directly can be used for crop growth monitoring. And same was evaluated for different regression approaches. For example, you can see these are the regression approach which has been used, and this is the per model. So you can see the R-square value how they are varying. Most important is regression models are empirical, site-specific, and so if at all you have a robust ready-to-transfer model, that can be used to derive these biophysical products which can be used for by the domain experts for different purpose. So you can put them together, giving some weightage, and can develop a crop health index for crop growth monitoring or mapping or for site-specific nutrient management, as I told you. So the different districts map is given over here. So which district is doing good with respect to crop health or crop growth conditions? Accordingly, one can go for different management practices. Then coming back to the last phase, how spectral on-mixing one can do or identifying unknown spectra. So this is what the field of view, if you have within the field of view, you have the sensor's field of view you have. Like sensor is at 25 degree, you can know what is the field of view. So you have different feature, again at 1:1 ratio. That means 50% is the background, 50% is the target. So like this, you have 50-50, like you have a lawn, you have a tin sheets, you have a grass, you have a thatched roof. So you will get a spectra which will be mixed spectra. So you can have a pure spectra of a grass, pure spectra of tin, then you will get a mixed spectra now. So that's what happens. 1:3 now here. So what happens to the spectra now we'll see. For example, you see here. So you have a pure spectra of asbestos you can take using the spectrometer close to that. You can have pure spectra of a grass. Then if you put a sensor at certain height, you have a field of view on which the 50% is your asbestos and 50% is your grass, or 25% is your asbestos and 75% is your grass. You can adjust that. So that experiment was conducted, and you can see how the spectra is changing. Having a pure spectra of asbestos, having a pure spectra of grass, then having a mixed spectra, having different composition, you are able to see the spectral pattern, how they are changing. So now question is, if at all I have a spectra which is unknown to me, for example, this is a spectra which is unknown to me. You can go back to this is a spectra which is unknown to me. Then I want to identify what the spectra belongs to, what kind of target it has or what kind of combination it has. So one can go for spectral on-mixing using different approaches so that you can know what the target is. I think when you take in the field the spectral data, many times what you get is you get the noises in the field, as I told in 1400, 1900. And the lab conditions, you get the spectra, very pure spectra. So one has to go for noise corrections at 1400, 1900. For that, this method was developed using signal noise thresholding, and the weightage given is spectral angle mapper and spectral feature fitting and binary encoding, so that one can correct spectra. For example, you see here, this is the field spectra, this is the pure in last spectra. And using that method, one can correct the field spectra. This is what the field spectra corrected. This is the field spectra which has a noise. This is the pure spectra in the lab condition. This is the field spectra which got corrected. So this is kind of a correction technique for the field spectra, and which can be used for analysis. For example, this is a spectra which is unknown to me. I don't know the composition, for example. This is a spectra. I want to decompose that, do spectral on-mixing, and go for matching with my spectral library so that I can get what is the percentage. So I have a pure spectra of asbestos and also grass. So I can go for different kind of compositions of those. For example, 0% asbestos, 5% asbestos, and 95% grass, 10% asbestos, 90% grass. So like this, I can keep on changing. I got a simulated spectra. Then this spectral mixture, whatever the spectral library I got for different mixture combinations of the target is combination is asbestos and grass. Then I can go for spectral matching using the techniques called spectral angle mapping, spectral feature fitting, binary encoding. Then based on the score, I can match the base spectra and I can identify which spectra looks to be good. So having an unknown spectra, you can use this technique to know what is my target is. So this is again another kind of applications. If at all you have a spectra of a feature which you don't know what is the combination, so if at all you have a end member of pure spectra, then you can identify. Same also can be done in the satellite data using Hyperion data. Same study was done. For example, this is what the pure spectra which is collected using Google Earth GPS data, point of a agricultural lands, bare lands, then urban features, then spectral angle mapper was used for classifying them. Then suppose you have a unknown spectra which is given over here, then I have a pure spectra. Based on that I simulated the spectral library, then I went for matching, and the matching tells me that the spectra which I have that corresponds to a pixel having 60% agriculture and 40% urban. So that's what the spectral matching can be done again using the same technique, SAM, SFF and binary encoding. I understand all these techniques has been discussed you before in the previous classes. So using that one can derive what is my target and what is my background. So these are some of the potential applications with respect to discrimination of the different varieties, crops, discrimination in the genotypes, then crop condition mapping, assessing or estimation of different parameters using empirical and biophysical parameters. So thank you very much.